So for this session, this is our last session, session 7. So we'll be covering the topic on multi-omics strategies and applications. And let us first start with the learning outcomes. You will be able, by the end of this session, to explain the concept of multi-omics and integrative multi-omics, to state the stages of multi-omics approaches, to define the steps of planning a multi-omics strategy, to list the tools used for analysis of integrative multi-omics datasets, and to comprehend the challenges of analysis of integrative multi-omics datasets. So before we talk about multi-omics, let us define the components of multi-omics or each omic separately. So the major omics types, you already studied with Professor Easter, the genomics. There are two genomics for short reads and for long reads. And these are all types of genomics. When we say short reads, that means we are analyzing the genome or exome, that is the study of the exons, using paired or unpaired reads. And this type of study started the study of the single variants within the genome. While using genomics for long reads, you are analyzing the repeated sequences or the repetitive sequences within which are long repeats within the genome. But at the end, you are studying the genome with different approaches, but with different aims. At the end, it's sequencing the genome and defining the genome content of a specific organism or a specific cell. Other type of omics is the epigenomics. And epigenomics is the analysis of DNA methylation, for example. And these are epigenetic modifications that takes place after the DNA replication within the cells. Transcriptomics, it's another type of omics, and it's analysis of gene expression. Regarding that means you are studying the messenger RNA and its transcription, frequency, type, mutations. All these are part of transcriptomics. Proteomics, as we already explained in the previous sessions about the different methodologies of analyzing proteins, structure, quantification, modification, post-translation modification, interaction. All these are part of proteomics. Glycomics and glycoinformatics, it's another type of post-translation modification of proteins, or maybe the type of carbohydrates that's not connected to proteins, but is studying the type of carbohydrates within the cell, and this is called glycoinformatics. Metabolomics and lipidomics, it's also another type of metabolites found within the cell, but it has its own science, its own ways of and technologies of studying this type of metabolites. Single cell RNA sequencing, this is part of the transcriptomics, but again, it is targeting certain type of cells. Could be cancer cells, or could be, for example, neurons. You are targeting a single type of cell, but it falls under transcriptomics. Spatially resolved transcriptomics, again, this is analysis of cells with respect to their spatial orientation. We mean by spatial orientation that means we are studying specific locations within tissues or cells, that studying either for example, in the plasma reticulum, or Golgi apparatus, or mitochondria. So we are oriented to a specific target of organelles, or maybe part of the cell within the tissues. So radiomics is the analysis of medical imaging. All these types are considered types of omics. And when you combine one or two, or maybe a number of these omics, then you are talking about multiomics approach. So it's a joint multiomics, a joint analysis and integration of two or more types of omics data. So what is multiomics? Multiomics is an analysis, is an integrative research approach that combines data from genomics, transcriptomics, as I mentioned, the different types of omics. You select the type of omics according to your research question, your objectives, and your expected output of the research. So, and this gives a holistic study, or holistic perspective about the cells you are studying, or the cell interaction, or maybe cell environment interaction. So compared to single omics approaches, multiomics analyzes multiple layers. That means from DNA, RNA, proteins, and metabolites. You are collecting a sets of data and integrate them to have more information, holistic information about what you are studying. So the multiomics approach allows researchers to gain a comprehensive understanding of the dynamic relationships and regulatory mechanisms among molecules. So the aim of multiomics is to identify key molecular features and uncover genes, regulatory factors, and metabolic pathways that play central roles in complex biological states. So thereby advancing the understanding of how biological systems operate. So it's very important to study multiomics at using different omic techniques. So multiomics concepts, as you can see from the drawing, you use the different technologies that is specific to a specific type of omic, like genomics, epigenomics, transcriptomics, and proteomics, and metabolomics. And so when you're studying each type of omic, definitely this is not enough information to give you more or holistic insight about a certain type of tissue or cell, or certain type of protein. We might be studying a protein from its gene level up to its interaction level. So you need to study different type of omics to combine data from different approaches of omics, from genomics up to proteomics or metabolomics. And then you study the interaction between these different layers, or the different data sets, and this gives you more insight about the network, or how the pathway of these metabolites has been coded within the genes, how they are transcribed, how they are translated. Is there any process of post-translation modification? If there is any process of interaction with other proteins? So all this information, you gather it, and it will give you more insight about about a specific protein, for example, you are studying. And this will help in defining biomarkers and targeted therapies. And this is now used as a science. Multiomics is now used as a science or approach to define what we call precision medicine. and even in the environment, in the plant, in the plant or agriculture field, is widely used to improve the production of plants, to define certain type of proteins or metabolites that could benefit the environment and the growth of microorganisms in certain environments, and so on. All these are needed to study a holistic study, starting from the genes up to their how they are functioning and taking part within the cell. So when you design a study for multiomics, definitely you need to consider other non-omic data. So when we say non-omic data, that means other environmental and environmental exposure factors, other demographic data like age, like economic parameters, dimensions, and so on. So other non-omic data you need to integrate in your study, and this is really one of the challenges. You need to define these external factors and how they affect which layer of this multi-omic or omic approaches is greatly affected by external factors. Usually, different researches, they tend to study the non-omic factors that affect on genomes as the first step of studying the first layer of multi-omic studies. We are going to speak more about what type of sample we need to, or how to process the samples. There are different ways of processing samples, either in bulk, that means you are studying the whole tissue without or maybe studying a whole collection of microorganisms in an environment without targeting specific organism, or maybe studying a whole part of a plant, like a stem or a leaf, without targeting specific cells within or specific parts within these parts of an organism or a plant or an environment. So this we call bulk. And single, that means you are targeting a single cell, a specific cell. For example, in human, you are targeting a neuron, you are targeting a specific cancer, cancer cells within a tumor, or maybe targeting a lymphocyte, or maybe in a plant, you are targeting a specific for example, specific leaf cells, or maybe root cells, and so on. So spatial, that means we are targeting a specific part within the tissue, or a specific location within the cell. So these are the different approaches of how dealing with the sample, and we are going to see on a slide on this. So the stages of multi-omics analysis, or how you process from sampling, data collection, up to the getting the results on the analysis, there are four major stages. The first stage is data acquisition, and of course, this depends on the type of omics that is related to your study or your research question. You could be combining genomics and transcriptomic, and that's it, or maybe combining genomic, transcriptomic, and proteomic, and this is another way of it's considered again a multi-omic study. And then after data acquisition, then you have data pre-processing. That means you are making control checkpoints for your data. And then normalization, noise reduction. When we say noise reduction, that means, for example, if you are using different type of sets of data from genomics and proteomics. Definitely, you need to reduce any errors. That you need to reduce any errors. That's mean the reliability and validity of your data. So, and this has different methods according, it depends on the type of your study. So, and then we come to the third stage, which is data integration. You want to integrate all these data sets from different experiments into a single set. So there are now different softwares, there are different models available in the internet. That is one of these. All these models are using statistical packages. It depends on what you want, on what data you have, and how these models or these softwares accept this data. You might need a coding system. You might need a specific way of entering the data. But again, you select the model you are used to integrate these data sets according to what type of data you have. Is it genomic? Is it proteomic? Is it transcriptomic? And so on. So these type of softwares are usually guided. And definitely when you start working on multiomics or planning your multiomic study, you will make a flow chart of your plan or your process or your methodology, and you select accordingly what sort of software according that is related to your data sets and related to your outcome. So all of these are using statistical analysis packages related to the type of data you are using and your outcome. And then the fourth stage is the data analysis and interpretation. And this is mostly used by informatics and again, statistical techniques, algorithms, and so on. It gives you more insight on biological pathways. It gives more enrichment on your what you should do. It gives you a significance of your study, and so on. So the pipeline for multi-omics data, as you can see, it starts from sampling, and then we collect the data from the different samples using different technologies, and then we transform this data interpretation. These are all at the level of the omics level. So we collect all this data and then we process it or integrate it as multi-omics. And then there is normalization of this data, quality assurance, multi-omics databases. There are different databases now for multi-omics. And then you can, this is one of the steps of how to validate your data sets, and and if there are any errors, you can go back and start where the error is it target to a specific omic, or is it target to the integration? So this is the pipeline, how it goes up to you end up with a model for your results, and this will guide you for any experimental work you want to apply. You might come up with identifying a certain protein that can be used as a biomarker or as a therapy for certain diseases. And so this will help greatly in defining your second step for experimental work at the clinical level, at the environmental level, or maybe in further in vitro experiments. So this is, as I mentioned before, there are different approaches of sampling for multi-omics. Either you are studying bulk, and this we call it bulk sequencing methods. That means you are using a different type of cells. Here, the different type of cells are colored in different colors: blue, green, and yellow, orange, and red. Which you study can be studied in each cell type, but only an average value per sample. For example, you are studying a protein. You have different type of cells, but you are targeting a specific protein. Okay, so you are studying this protein in different types of cells in each cell type, but at the end, you are taking an average of all these type of cells. So it's a type. That's why we call it bulk, because you are taking an average from all type of cells. The other type of sampling approach is single cell technologies. And this allows a study of single cell studies, and then you are targeting a specific type of cells, and study a certain protein. You can study the different levels of multiomics, from genomics up to the transcriptomics or metabolomics. But here you are targeting a single type of cell. Spatial, as I mentioned, it uses the interaction between cells, and you are targeting a specific location within a tissue or a cell. So that means you are targeting an organelle or a specific, maybe a cell membrane, or maybe if it's a tissue, maybe a certain type of or certain location within the tissue. Especially, for example, in this study, they are working on cancer cells. So they are targeting certain type of cells within a tissue. They are taking a tissue, part of the tissue, and a certain location within the tissue they are studying. So this, we call it spatial omics. So why do we need to study or use the approach of multi-omics? There are, of course, different benefits. It gives the researcher more understanding of the complex biological processes. It gives more insight into the study of rare and difficult to understand diseases. And when we say disease, we mean in human, in any organism. It could be a plant, it could be insects, it could all this type of organisms or file level. Then you can use multi-omics to understand the different diseases. It gives more insight about the population, genetic makeup, proteome makeup, and so on. So at the end, it gives the most important benefit is the study of single molecules that could benefit, be part of precision medicine. Could be drugs, it could be vaccines, it could be biological or supplemental ingredients for food. It could be supplemental ingredients for fishes, for organisms in different environments. So there are a lot of benefits of studying or using the approach multi-omics to study certain metabolites or certain molecules within a cell, for the benefit of the population you are studying, and for the benefit for the bigger society. That is means for the benefit of the interaction between the different communities, if it's a human, if it's a microbe, or even human-plant interaction. Here is a type of combinations, different type of studies. As you can see from the table here, you can combine genomics and transcriptomics. Again, it depends on your research question and your target. or you can combine genomics and proteomics, genomics, epigenomics. So these different type of studies in the table shows the implications. The implications depends on your research questions. So at the end, it is considered a multi-omic study or research. Or you can combine three omics studies, or four, or all the type of omics. But definitely it needs, it's a big project. It needs time. It needs cost. So you plan your study according your time factor, your budget, and your expected outcome. What are you looking for, and your research question in the first phase. So integrative multi-omics, that's it's interchangeable terminologies for either we call it multi-omics or integrative multi-omics. But integrative, it's meant that you are using more than two omic processes or approaches in studying specific molecule. So you are integrating different omic processes, data sets to reach a specific outcome. So integrative multiomics has the potential to improve disease diagnosis and treatment by uncovering underlying molecular pathways. So because you are studying from the genome level, as here in the study, genome, proteome, metabolome, and transcriptome. So all these data are integrated into one analysis setup of data sets, and to give more insight. Because if you want to talk about production of vaccines or certain type proteins to be used as supplemental molecules, then definitely you need to study the whole process of production of these molecules within the cell. You cannot confine your study for a specific type of omics. So again, it depends on your research question. So these type of studies, they give more approaches about the pathology of these type of molecules, the interaction with other proteins or other metabolites within the cell. So you need to have a holistic study to understand better and in depth the molecule you are targeting to use as the drug therapy or biomarker or as a supplement. Again, it's the same. It's just more. This diagram is more elaborative, showing the different methods that can be used for omic technologies. But again, the process is the same. Start with sampling, and then at the level of the sample, how you process it, the type of data, and then how you combine this data into different sets of. You have different sets of data, and how you combine them from different technologies, and then you integrate them. The most important, you need to validate the data, which is definitely the use of controls, the use of validation methods, statistical analysis to test your data, reliability, validity, and so on. So, so you either have horizontal integration or vertical integration. Horizontal integration, we means that you are studying cells. You have different type of cells, but studying for example, omics as a different type of cells, and getting the data from the different types of cells at the level of proteomics. While vertical, you are using the different type of omics and integrating the data from the different type of omics vertically. That means in different, for example, genomics, proteomics, and metabolomics. Again, this is another way of using a framework. Here in this drawing, it's the newly introduced concept is the use of AI in multiomics. After collecting the data and doing the normalization and defining what feature you are looking for, then all this now is further analyzed by using models of artificial intelligence. And as you know, the models, which is the deep learning models in the artificial intelligence, is either supervised or unsupervised learning. And when we say supervised, that means you already trained your AI agent into specific analysis of certain features. While unsupervised, that means they are not trained, but they are left to these AI agents to identify the or to analyze the data sets without having been trained on certain features. So this is the difference between the use of different type of AI approaches, but this is a new revolution into the multi-omics approaches. And in fact, it gave more. Now studies are talking a lot about the use of AI in analyzing multi-omics because it's using a different algorithm, and this is combined with statistical analysis in the different softwares or databases. It gives more insight about the data sets and more information about the outcome of these studies. This is an example of a study. They are studying different type of cancer cells. As you can see here, they are using different type of cells. They are collecting data horizontally from genomics, but at the same time, they are studying vertically. So vertical data integration. That means they are using genomics, epigenomics, and transcriptomics and proteomics. So what we mean by horizontal and vertical, it depends again about what is your study, the question, the research question. But this is what we mean about horizontal and vertical data integration. So even if you are using different cells and using one omic approach, it's also considered as one omic approach. So but the benefit is that you are using the integration both of the horizontal data with the vertical data, and this is very important for studying a specific type of molecules, to analyze it to a step where you can use it as a biomarker or as a drug or a supplement for any type of environment you are studying. So again, multi-omics strategies for personalized medicine is another way of another way of showing the benefit of multi-omic studies. So here in this study, they are using different types of approaches, phenotypic data, multi-omics data, and single cell spatial. So these type of different approaches, it gives more insight about their studying at the level of because when we say personalized medicine, that means there are specific variation between different type of populations. So you need to define certain drugs or certain biomarkers for specific type of population. And this is now it's very high. It's in very high demand for personalized medicine. Um, because there is a difference, wide difference between drug interaction in different populations, and even the type of biomarkers that is specific to a certain disease or specific to a certain character within a certain population. So it's very important now multiomics for studying and for the use in precision medicine. And as you can see here, artificial intelligence became as a parcel and part of this analysis of data. This is another way of also a type of study. They're starting from genomics, transcriptomics, proteomics. They're collecting all this data, and it in a model what is called integrative also analysis. And here they are studying melanoma patients. This study is meant to managing this disease, and they managed to analyze this data using bioinformatics and to identify biomarkers, and this again helped in what we call precision medicine. This another way of multiomics use in plant breeding efficiency. Again, they started with the metabolomic, proteomic, and transcriptomic genomics, and they were able to define certain type of metabolites that can be used to accelerate crop improvement, production, and breeding. And so it facilitated the breeding, increasing the genetic, diversifying the genetics of these plants for the betterment and production of the plants. So I'm just showing these type of examples to show that it is can be used wherever it is needed to improve and to for the benefit of the population or study, whether plant, whether human, whether microorganisms. Again, this is identifying another study, identifying or characterizing the biological elements in crop traits, and they are using these crop traits for the benefit of other crops and the improvement of other crops traits. Like for example, if you want to increase the resistance of certain plants against insects, so you have specific trait in certain plants, and you can use by genetic engineering to improve other crops against certain insects. There is this table shows different analytical methods. I'm showing this table just for knowledge. So there are different tools for analyzing for helping to integration the data, because as we mentioned, as we saw before, the stages, the 4 stages of multiomics. Before analysis, there is integration of this data. So these are the different types of tools or softwares that are available to integrate your data. It depends on your study. For example, the first one, which is used for integrating epigenomic, genomic, and proteomic and transcriptomic data. If you have your study has this type of data. So others, they don't do that. For example, the other one, it combines only the epigenomic and the transcriptomic. It's not available for genomics. So you can select what type of tool, and it is guided. And I always recommend for my students to so that if they want to use multi-omic approach, they need to know the R language, or it's similar to what we call Python language, because now these software, they use the R analysis approach, which is a specific language, the coding language for these softwares. So this is very important for nowadays, for all types of packages that is used in softwares. Before you dive into any study or analysis, you need to know what type of language the different softwares use. But most of them, they are using the R coding system or the R language. Again, this is it shows it's just a translation of the previous table in a visual form, the different tools. And the different tools, it depends. For example, if you want to do your study targets disease subtyping, then you need to use, for example, iCluster, because it uses different statistical methods. So all these are different statistical methods: Bayesian, multivariate, fusion. These are all different statistical packages. Again, you select the software or the package according to what are you targeting, what type of experiment you are using. Is it bulk? Is it single cell? Is it spatial? Okay, is it horizontal data or is it vertical data? So you have to select your type, or the tool you are using to integrate your data sets, depending on your data, your target. Are you targeting to study the subtyping of the diseases? Is just biomarker prediction? Then you will use different statistical package, and so on. So these are the different tools for integration. So nowadays, the most popular integration tools are used is the multi-omics factor analysis, which is MOF, and the iCluster. These are widely used and widely tested. So each one, it has its input, and each one, it has its output. So for example, for the iCluster, it uses a different framework or statistical package, with the Bayesian framework, and it's a specific package. But the most important for these two is that they are able to integrate data from genomics, transcriptomics, epigenomics. So they have a wide array of data they can integrate, unlike the other tools. So for the iCluster, it is widely used, and it is very suitable for clustering analysis and molecular subtyping and identification of your data. For the multi-omics factor analysis, MOFA, it's again, it uses different statistical analysis package, but different logarithm, but it's widely used if you are targeting or you want to study biological processes, identifying pathways within cells. So it depends on your data and your expected output. Usually, researchers, they combine both in studying the identification, and at the same time studying the biological processes within the cells. So there are different way of using different algorithm and statistical analysis for the two sets, but these are the two widely used nowadays in research. Why? Because they accommodate different type of data sets from genomics, proteomics, and transcriptomics. So they give more space for researchers to combine different omics in a single data set. This is just a table for you, just to see what's the difference between iCluster and MOFA, which is widely used nowadays in researches, but it's just an orientation table. You don't need to know it in heart. It's in case if you want to use multiomics, you need to know what type of integration tool you want to use. As I mentioned, it depends on your input and your expected output. What are you studying? Again, according to your research question. The most important of this is the steps how to plan your multi-omic strategy. So I tried to put it in 10 steps. So if you want to plan to use multi-omics, what do you need? How do you move on with your research? First of all, you need to define your research question. And according to your research question, you define your methodology. And this part of this methodology, definitely your what type of omics you want to use. That is means the omic layers you want to study, and then define your experimental design, how you are going, for example, if you want to use genomics, transcriptomics, and proteomics, and metabolomics, how you are going to design your study to collect the data that answers your or fulfill your objectives and answer later your research question. So you select assays and platforms. For example, do you want to use bulk, single cell, or you want to use spatial, for example, targeted versus untargeted locations within the tissues? And then you plan your sample splitting. For example, as I mentioned, as I showed before in the cancer cells study, they are using vertical data and horizontal data collection. So what approach you are using? So this will help you to define what sample you need to collect and how you split them. After you do the analysis, after you collect your data, that means you do your study omics, different type of omics. Then you need to normalize pre-process for each omic layer. That means normalize, that's reducing the noise. That means having a control for each study, testing the validity of your experimental tests you did, the reliability of your test. That means you do pre-testing your study before you move on to integrate the data sets. So and then you select the integration strategy, again according to what is your research question and what we expect out of this study. Are you identifying cell types? That means you want to use iCluster. If you are studying biological processes, then you need to use multi-omics factor analysis, because these are different types using different statistical analysis, using different tools of integration. So, and then you integrate all this. After you integrate, you need to interpret your results and validate your results. That means if you studied protein, for example, how do you validate it? That means you use, for example, Western blotting. That means this is a protein you are targeting, and or you might use modeling, okay, or you might use to quantify. It depends on your research question. So you need to validate your output, and this is the different 10 stages how you move on with your multi-omics strategy, and this is very important at the beginning of your research. Then this diagram shows you how to integrate omics and non-omics data integration. So, as I mentioned before, non-omic data is very important. There are challenges in integrating non-omic data, and as I mentioned before, non-omic data it consists of demographical data, epidemiological data you are collecting beside the omic data, which is you already integrated and you studied using the different integration tools. So integration depends on using statistical analysis, definitely. After you get your omics data, then you need to study the integration of omic data, non-omic data into your data sets. And these have different softwares. But again, collecting the non-omic data, it has its own challenges. There is, of course, high heterogeneity in the medical data. If you are relying on medical records, there is high complexity within this data, especially if you are, for example, studying the effect of temperature on genes and this gene on the expression level and so on. So you are combining non omic data with omic data, and this it needs specific and very careful studies and having controls and studying at different levels, different concentrations, and so on. So there are different models, but these are all under test. So, but I'm saying that you need to consider the non-omic data on your studies. There are different challenges for the integration. Of course, the as you know, in cells, choosing your omic in the cells itself, there are the gene expression is is uh, is controlled by certain factors, and even the replication process of the DNA and the replication of genes, there could be mutations. So it's very difficult to if you are studying different cells, then it could lead different. It gives you different data sets, and this is one of the challenges of multiomics. So always you need to look into horizontal studies and vertical studies to accommodate any changes within the cells. Other things you need to consider, you need to select the proper statistical package that is related to the properties of each omic layer. And this is very important in the normalization level. The other thing is that there are dimensionality reduction. That means there are in each omic there is a space of analysis. We call it a space of analysis. That means within this space you are analyzing a specific type of, for example, protein. You need to know what is the dimensions or the factors that affect this protein within this cell, because otherwise within different cells you might get different data set, which are why there is a wide difference between them. And that's what we need to reduce the dimension. That's what we call the dimensionality reduction. We need to reduce these differences, and this is what we call how to optimize your experiment to reduce this diversity or the dimensions in that appear in the resulting in variety of the data sets. So these are all you need to consider before you embark into your experiments and into your multi-omic studies. So these are the main challenges. But one of the main challenges is the incongruent analytes. What do we mean by incongruent analytes? It's in multiomics integration, this problem arises when molecular features measured across different omic layers. For example, you are studying genes, transcripts, proteins, metabolites, and when you and then while you are studying this, you recognize that they do not correspond to each other. So this is what we call incongruent analytes. That means in fact, in reality, they should be coherency in your study, from genes up to from replication up to transcription and to translation. This is actually the fact, but sometimes this does not go this way. So you found they are not corresponding to each other, and this happens in many types of studies already addressed. So this type of discrepancy happens from in this pathway. It stems from the biological processes such as alternative splicing. As you know, in transcription, there is what we call alternative splicing. It happens from post-translation modifications. So all this affect the coherency of, or the linear process to goes from gene up to translation. So in this case, you it's an evident challenge, but you need to put it in mind and consider that this could happen, especially if you are working on eukaryotic cells. This type of challenges might reduce in prokaryotic cells, because, as you know, there is no splicing and there is no exons and introns. I don't mean the presence of exons and introns, but it's reduced in prokaryotic cells. So consequently, a single gene may produce many transcripts and protein isoforms, while a single metabolite may be influenced by multiple enzymes, creating a complex many-to-many mapping that prevents clean alignment across datasets and violates assumptions of direct correspondence in integrative models. So this is an evident problem, but you need to cater for it, and it depends on your study. So consequences of this is that false correlation, loss of biological signal, incorrect inference, mask temporal dynamics, that means mismatch time scales. That means if you expect that the gene expresses the frequency of expression is already recorded, then you might come out that this is not fixed as a time factor. There is dominance by one omic layer, unreliable pathway enrichment, inability to validate mechanisms. There is lack of direct analyte mapping to prevent experimental follow-up. So these are the main challenges. So this is the end of this session. Thank you. Do you have any question for this part? Can you hear me? Hello?
Yes,
Yes, we can.
Do you have any question for this part?
No, for me, no.
So this is the end of this course. I wish you all the best for your exam. Make sure to answer the questions I provided. It will really help you much. And be specific on your answers. Don't dive on unneeded information. read the questions carefully, and plan your answer also carefully. Structure it in a sequential way, and be specific and concise. So all this, after all this, I wish you all the best. If you don't have anything, then